Welcome to the 2020 United Mitochondrial Disease Foundation's Big Pitch. I am Brian Harman, President and CEO of UMDF, and we're delighted to present to you today the finalists of our 2020 Accelerators Prize, a $50,000 prize awarded to the top postdoctoral researcher in mitochondrial disease research. We launched our Accelerators program in 2019 to create a confluence of fast science and engaged philanthropy. The program gives our generous donor community a voice in funding mitochondrial disease research. When our donors give $500 or more cumulatively in a year, they unlock Accelerators benefits. No matter how they give, whether it's through a special event, a designated fund, or as a tribute to a patient, when they reach that accelerator level, they unlock these special benefits and join other engaged philanthropists, to get a firsthand look at the promising ideas being developed in mitochondrial disease research. In a little bit, over 500 accelerators will have the opportunity to cast their vote for this year's prize winner. And if you're not an accelerator yet, I encourage you to visit umdf.org/accelerators to learn a little bit more about the program and to join us today. There's still time to join so that you can cast your vote for this year's award winner. At UMDF, we aim to fund the best science. No matter where it is found in the world, we want to unlock discoveries for the patient families we serve. And to that end, our organization since 1996 has funded over $12 million in research projects across 120 plus labs around the globe and has led to $150 million in follow-on funding. We have done this with our legion of supporters for nearly 25 years. And we aim to fund researchers throughout all stages of their career. We have a prize for an experienced principal investigator. We have a prize for an early stage principal investigator. And today we're applying the finishing touches to our Accelerators Prize, a prize dedicated to promising postdoctoral research fellows. Today's presentations and these presenters will have the opportunity to join our inaugural winner, Dr. Arwen Gao, as an Accelerators Prize recipient. So let me tell you a little bit about how this works. UMDF has earmarked the prize at $50,000 for this year. And of course, that cannot be done without the generous support of our donors across the globe. Grant applications were submitted by promising postdoctoral fellows. And these applications are reviewed by our Scientific and Medical Advisory Board. A special committee of that board reviews these applications. And then today you're going to hear the 5-minute fast pitches from these finalists. They'll have an opportunity to share their project and ultimately how this impacts our patient families. Then every qualifying accelerator has the opportunity to cast one vote for the project they feel most passionate about. And then ultimately the prize is awarded to one of these finalists today. And here to share a little bit more about the selection process and to introduce our finalists is UMDF Science and Alliance Officer, Dr. Phil Yeske. Phil?
Yeah, thanks, Brian. Pleasure to be here today. Really excited to hear the presentations that are coming up. Wanted to start by sharing just a few more details about the process that we go through in the accelerators overall process. I'm going to focus on the 2 steps that are in the green box here, right? The submission of the grants and the review of the grants. And really, this is about establishing scientific credibility. When Brian approached me about this program a year ago and talked about donor engagement, I really got excited about it, but I told him we need to maintain our credibility as an organization, that scientific peer review is at the foundation of any award we make. So we co-developed this process. It really plays off both sides of donor engagement and scientific peer review. So as Brian laid out the process, to this year, in this cycle, when we posted a request for proposals back in January of this year, we received 7 submissions from postdoctoral fellows around the world, and that's for 4 countries represented. At that point, our scientific and medical advisory board, together with me, go through what's called a triage process, just to make sure, are those relevant to the UMDF mission? Are these applications that merit, based on a quick review, further peer review? So from that 7, we selected 6 to continue on to peer review. And this is the point where we really leverage our contacts within the Scientific and Medical Advisory Board and select outside experts, just to make sure we're getting the perspective necessary to review any of the proposals that are submitted. So each of those submissions has 2 sets of eyes on it. So 2 reviews on each of the 6, 12 reviews overall. Then we bring all that information inside. And once again, the chair of the SMAB, Dr. Bruce Cohen at Akron Children's Hospital, and myself go through that and really start to look at the ranking of the proposals to make a selection for, in this case, who should be the finalists. Traditionally, with a prize, we would pick 1. But in the case of the accelerators, we're trying to find a set that represent what we think is the best of the best science, and that our donor community, our patient community, will really relate to as well. So this year we selected 2 submissions, and you're going to hear from each of the postdoctoral fellows that submitted those in just a couple minutes. But before we move on to their presentations, I'd like to just take a second to talk about a central theme in their 2 proposals. And this is completely by chance. We didn't plan it this way, but their proposals have a central theme of heteroplasmy. And as we prepared for this, we recognized that that may be a scientific concept that you're not familiar with. And so I thought I'd just take a couple of minutes to talk about heteroplasmy, sort of set the stage for the speakers that are coming up. And you may hear some of this same language again. I think that's fine, because that helps to reinforce, and maybe you've heard it described in a couple different ways. So again, it really begins with 2 different types of DNA that we have, or 2 different places for DNA in our body. There's nuclear DNA and there's mitochondrial DNA. And of course, the nuclear DNA is inside the nucleus. And most of us learned about that in biology at some point in school. These are the chromosomes that really determine who we are, what we look like, these sorts of things. And there's really 1 copy per cell. The difference is for mitochondrial DNA, which only represents about 1% of the overall DNA content in our body. So 99% nuclear DNA, much larger, a lot more information, 1% mitochondrial DNA. The major difference is there are multiple copies of that mitochondrial DNA inside each mitochondria. Right? And there's reasons for this relating to bioenergetics and a lot deeper science. But for the purposes of today, what I really want to get across is we're going to focus on mitochondrial DNA, often abbreviated as mtDNA. So you'll see that on the next slide as well. And there's many copies per cell. So in a healthy cell, you would hope that all these copies of the mitochondrial DNA, which are maternally inherited, only come from 1 parent. Nuclear DNA comes from both parents, paternal and maternal. So only from the maternal side, you would hope that all of these DNA copies are identical. And in this cartoon, they're represented that way. But that doesn't necessarily represent real world. So that's when we get to heteroplasmy. So bear with me as I kind of talk through this cartoon and the various aspects of it. And I've tried to label each of the key components of it. But now we're focusing on just the DNA content within any given cell. So these are 3 cartoons of 3 different cells in different states of health. And that purple circle is the nucleus. And inside that is where the chromosomes would reside. I only had room to put 1 in there. But you'll notice that across these cartoons, that's not changing. Right? That chromosome remains as it is. That's nuclear DNA. Mitochondrial DNA are represented by these small circles. And we have 2 different types. So in blue are what are known as wild type mitochondrial DNA genome. And if we're translating that to lay language, healthy mitochondria, right? No mutations, exactly as expected. In red, we see mutant mitochondrial DNA genome, and these are disease causing. So that means something has gone wrong in the machinery when the DNA was being created for the mitochondrial DNA, and a mutation occurred. And so those red circles are different than the blue circles. And it may be a very small change, right? A very small point inside of a long chain of nucleotides. But that one change can have a drastic effect and lead to mitochondrial disease. So what we're seeing across the screen on the left is a healthy cell, and you may be surprised to see a red circle in there. What it tells us is cells in the human body can actually tolerate some level of having mutations inside there, so copies of the DNA that are not identical. And so this mix of healthy wild-type mitochondria to mutant mitochondria is what is known as heteroplasmy. And there are a lot of analogies you could use for this, and I think our finalists will probably present their own. Sometimes I think about like cholesterol, right? We're able to tolerate a certain level of cholesterol in our bloodstream, but when it hits a certain point, all right, when it gets too high, that's when cardiac disease could arise. Well, you might think of it the same way here, right? There's a certain amount of mutations that naturally occur and are tolerated, but as they increase in number, or if they're naturally formed in higher number, as you see in this center cell or on the far right, that's when problems can arise from a disease perspective. So what we see are 3 cells in 3 states of heteroplasmy: very low heteroplasmy, normal function, chances are no symptoms for the person with this type of cell or this type of arrangement. In the center, we see that middle case where there's some amount of heteroplasmy and we begin to get abnormal function. So the mitochondria are not able to overcome these mutations and perform exactly as they're supposed to. There may be some symptoms associated with that. And then the far right is the most severe form of heteroplasmy, the highest level of heteroplasmy, if you will. And that's when we have the highest level of mutated mitochondrial DNA relative to the amount of healthy, represented by the blue. So this is when the most severe symptoms can arise. And for mitochondrial DNA, forms of mitochondrial disease can sometimes be lethal. So there's a lot of subtlety and nuances to this, but suffice it to say, there's some threshold where disease begins to set in, and that threshold can change. Maybe it's when only one red circle here goes to 2. If it's a tissue type that is highly dependent on the mitochondria to make a lot of energy, think of the brain. The brain requires the most energy, right, in our body. So for the brain, even a low level of heteroplasmy can be a challenge to maintain normal function. Other types of tissues or other mutations may be able to tolerate a much higher level of heteroplasmy. So that's this first bullet point here. The take-home message from this overall slide for you today, as we begin to hear from our finalists, is from a research perspective, which is why we're here. UMDF, its research mission, our desire to find the best science in the world, no matter where it is, today has a very central theme to it, and that's heteroplasmy. And the question that's being asked, and the submissions we've received have proposed ways to address this: how do we potentially drive heteroplasmy back in the other direction towards healthy cells? That is to say, from a higher level state of heteroplasmy to a low level state of heteroplasmy. And in doing so, the hope is that the symptoms of the disease reduce with it, potentially mitigating them to the point where a patient might be able to live a normal life without the symptoms associated with mitochondrial disease. So that's my quick overview of heteroplasmy. Again, our 2 speakers coming up will probably speak to this as well. I hope you found it useful. But at this point, we're going to move right into our first presentation. I'm pleased to introduce Dr. Lia Mayorga from IHEM Institute in Argentina. So Lia, please, we look forward to it. You have 5 minutes.
Okay, thank you so much for the presentation and the opportunity. I'm going to start by telling you a story of a patient of mine. This is Luis and his mom, Anna. Luis is 12 years old. He's had 3 strokes in his life. He has seizures and trouble walking. He was diagnosed with Mira syndrome, a mitochondrial disease, a genetic disorder due to a mutation in his mitochondrial DNA. This is what a cell from Luis's body could look like. You see, most of his DNA that is in the nucleus is just fine. His problem is in a tiny DNA molecule inside the mitochondria that's essential for the proper functioning of the organelle. This is a cell from his mom, Anna, a healthy adult. Luis inherited his mitochondrial DNA from his mother. So you see, his problem is a matter of proportion of unhealthy mitochondria or heteroplasmy. So if we could change Luis's cells and make them look more like his mom's cells, well, that would be a great strategy. And that's what's called heteroplasmy shift. Now, why hasn't this master plan reached the clinic yet? Well, mitochondria are pretty picky as to what they let in. So other gene therapy strategies that have reached the clinic for nuclear genes won't yet for mitochondrial genes. So we propose a totally different approach, taking advantage of the cell's own machinery and communication between the nucleus and mitochondria, using an FDA-approved drug that acts in the nucleus and could help the cell decide which mitochondria to toss and which to keep to take over. My name is Lia Mayorga. I'm from Argentina. I'm a pediatrician that did my residency in this wonderful hospital in Buenos Aires, where I got to know patients with mitochondrial disorders. And then I decided to cross over to the bench side and got into research in my hometown, Mendoza, where I continue to do research with this amazing team and also take care of patients. So what does this title mean? What is the epigenome and how can it impact mitochondrial heteroplasmy? We'll try to make it simple. Imagine a cell is a small company. The nucleus is the CEO, and he has a bunch of workers, among them mitochondria, powerhouse of the cell, making lots of energy, and they communicate with the nucleus. Now, this CEO can have different ideas as to how he manages the company. Ideas or marks in the DNA, epigenetic marks, that can radically change the cell's functioning. So it's the same guy, it's the same DNA sequence, just different marks, different ideas, different epigenome that can change over time and under different circumstances. Now, when the cell's going fine with hard workers, no problem. Well, when a couple of lazy workers come in the company, the cell norm says to shut it down. Why is this? Well, there's plenty of cells in the neighborhood that can take in the work, and there's no need for an inefficient company to keep on going. Now, when most of the workers are lazy or unhealthy, as in what happens in the high heteroplasmy cells that we saw in Luis's body, well, union makes force, and they give weird ideas to the CEO and force him to maintain the company open. So there's an epigenetic pro-survival patterning that keeps these high heteroplasmy cells going in an unhealthy way. So if we could change these ideas, or maybe get rid of them, these cells would definitely shut down and give room for the healthier cells to take over. This is what a tissue from Luis's body could look like. You see, most of his some cells have high heteroplasmy and others low heteroplasmy. These high heteroplasmy cells have these pro-survival epigenetic marks we were talking about. Now, we've done some in vitro work with promising results using azacitidine, which is a general epigenetic modulator. And what it does is erase these epigenetic marks. And we've seen that these cells shut down and give room for the healthier cells and heteroplasmy shift. Now, azacitidine is an FDA-approved drug, which means a shortcut to clinical trials. So we'll definitely pursue this line of research. And next, we want to see if we can modulate more specifically the epigenome. For that, we plan to study in depth these high heteroplasmy cells and really look into these epigenetic marks to see if the black, the blue, or maybe the yellow is the most important epigenetic pro-survival mark, and in this way, aim for a more specific target for treatment. So I hope I've convinced you that modulating the epigenome is a great idea for heteroplasmy shift and a winner proposal, so that together we can achieve our ultimate goal, that is to help kids like Luis. Thank you so much. I want to thank my mentor, collaborators that believed in me and in this project, and especially the UMDF donors for this unique opportunity.
Great.
Thank you.
Thank you, Lia. That was a great presentation. Appreciate you doing that. We'll now move on to our second finalist, Dr. Kinsley Belle from Stanford University in California. Kinsley.
Thank you. Today I'm going to talk to you all about my research investigating mitochondrial disease and heteroplasmy. So first, a little bit about me. I've always been interested in using science and technology to improve the human condition, But this project actually became personal for me when I started to interact with the first patient that I enrolled. We had actually many similarities in our life, our age, our academic pursuits, the movies and TV shows we liked. But I was really able to see the seriousness of mitochondrial disease firsthand. And this remains a motivation for me, as well as the people in my lab. It also helped cement as a career goal in the future to become an independent researcher in the mitochondrial space. This is because I feel that there's a lot of work that still needs to be done, and that mitochondria actually have a wide implications of many diseases as well as aging. So why should we investigate mitochondrial heteroplasmy? Just to reiterate, heteroplasmy is the fraction of a diseased or mutated mitochondria chromosome within a cell. So we all know approximately 1 in 3,000 of us are affected by mitochondrial disease. However, every 1 in 200 of us carry some form of deleterious mutation, usually at low heteroplasmy levels. If you look at the panel on the left, let's say that you're one in these 200 people. You may have, low in the 20s to 50% heteroplasmy, but as the mitochondria in this cell divide, as the cell divides, you may increase these levels. And if you look at the tissue level, where you may have just a little bit of disease cells, as they expand, the whole tissue may become more disease, and that will correlate with disease severity. We look at this from an inheritable and development standpoint. We get our mitochondrial DNA from the mother. We start as a glob of cells. Those cells proliferate, and we mature into a body. So the cells which actually harbored this mutation will be indicative of where in the body we actually see the heteroplasmy. And as time increases, this heteroplasmy may also increase. So overall awareness of heteroplasmy is actually important for improved clinical diagnostics, as well as inventing and developing therapies and cures in the future. So my project and what I hope to find out can be split into 2 parts. The first part is us expanding our knowledge on disease. Why does heteroplasmy affect individuals and tissues differently? And there's the second part, the search for a cure. Can we find drugs, FDA-approved small molecules, or certain stimuli that actually improve patient conditions? So for the first project, what I really want you guys to understand, what's really important about what we've been doing, is that one of the current limitations in research is that you go into a clinic and you usually get 1 tissue sampled and you get a single time point of information. One thing there is that tissue may not be indicative of the disease in this patient. But in the patients in my study, we always take at least 3 samples. For example, if you look at panel A, I grabbed blood, skin, and urine from our patient and looked at heteroplasmy across these tissues, and we saw they were all different. I even looked at the single cell level and saw interesting patterns there. But another limitation is that you can't always study look at the heteroplasmy changes long term, which we do here. If you look at panel E, this is actually our ability in vitro to look at changes in heteroplasmy in the case of adding drugs, for example, and looking at see how heteroplasmy changes over time, which can't be done in the clinic. Lastly, I can model this with patient specific cells in almost any tissues. These experiments were actually done in patient-derived neurons and cardiac myocytes. As was stated before, cardiac myocytes and neurons have a high energy demand, and we can't take a patient biopsy of their brain or their heart without further damaging their health. And this project allows us to study these tissues. The second part of my project, like I said, is the search for a cure. I just want to reiterate, there are no cures for mitochondrial disease and not many reliable treatments for our suffering patients. So the cell lines I discussed earlier, what we look to do is expose them to decreased metabolic substrates and reduce oxygen levels to see if we can increase the efficiency in their metabolism, thus selecting against the bad or mutated mitochondria and increasing the healthy mitochondria. We also want to do this with FDA-approved small molecules. While some drugs have been shown to alleviate mitochondrial health or improve mitochondrial health in an acute method, we look to do more than this. We actually want to see if we can use these drugs to select against the bad mitochondria and just not supplying acute relief, but looking at the root of dysfunction and decreasing heteroplasmy overall. So just to kind of reiterate and summarize, the overall implications of my study is that we're more than just the classification and characterization of new mitochondrial mutations. Our studies and our methods highlight the importance of good diagnostic methods and identifying heteroplasmy across multiple tissues within our patient. We also look to lower heteroplasmy through selective pressures using FDA-approved drugs as well as changing the metabolic stimuli, such as reducing sugar or lower oxygen. And all this work that we've put together encompasses the establishment of a drug screening and research pipeline that can be scaled up to include more mitochondrial patients, not just the ones currently enrolled in our study. I'd like to thank first the families that are involved in my research, UMDF for giving me the opportunity to present my work, as well as my lab members and mentors for supporting me, and you, the audience, thank you for your time.
Great, thank you, Kinsley. Very, very interesting, excellent presentation as well. So, Brian, here we are.
Well, I got to say, as both a layperson and a donor to this wonderful organization, so great to see this explained this way in such a digestible way. So, thank you for donors like myself who crave this kind of information and want to understand where we can best put our donor dollars. Thank you so much. And something else that struck me on both presentations is our shared motivation, right? It's inspired by the patient families we serve. And I'm so grateful that not only did you show us really great science today, but you tied it back to what's most important to all of this, the patient families we serve. So I wish you both the best of luck as our voters now have until Sunday, June 28th at 6 o'clock Eastern to cast their vote. So for our Accelerators members, I ask you to check your email for voting instructions. And for those of you who may be itching to become Accelerators, to take advantage of this really unique opportunity and help us accelerate really great research for the next generation of mitochondrial disease experts, I encourage you to visit umdf.org/accelerators to become a member today. You can sign up right up until that deadline on June 28th. So we encourage you to do so. Thank you so much, everyone, again, and go fast.
Thank you.